these algorithms.
Thank you for the introduction, and thanks a lot for the invitation. And thanks a lot, Christian and Marianne and Kuhn, for setting up this session and inviting us. So my name is Georg I'm from Medical University of Vienna, and in the next couple of minutes I'd like to discuss a bit with you the idea about establishing correspondences, about correspondences across individuals, across groups, across species, and how we can accommodate maybe different notions and different concepts of correspondences in order to link observations across observations. And so I think everybody here knows that the idea of registration, of kind of mapping individuals to a big kind of stable reference space in order to make observations comparable is an extremely successful concept in our community, and a lot of what we know about groups, a lot of what we know about the architecture of the brain, we know because we were able to kind of summarize our observations made across large numbers of individuals in a kind of unified reference space. And the general concept here is that we have a means to establish correspondence, like for example anatomy and the shape and the morphology of individuals, that allows us to map and deform individual spaces, individual brains to a common reference space, and to say, well, this position on this individual, that's actually the same position on the different individual, and then treat the observations that we make, like function and activation or connectivity, as features that happen on top of this kind of reference space in which we think. And I think there have been like a tremendous line of work that made this better and more stable and more accurate. So I think one of the later ones is by Emma Robinson, who also introduced the idea of integrating many sources to establish this correspondence in order to make it more stable and more accurate. And I think many of us are working with this kind of framework. So one question though is that when we are looking at the variability that we are observing on top of this framework, and this is a plot out of a paper by Sophia Müller, who looked at the variability of functional networks across individuals and realized that there are areas like the frontal cortex and the parietal frontal cortex that are fairly variable across individuals, whereas others like the motor cortex or vision are fairly similar. We realized that this variability might be a composition of different differences, like for example the actual differences of the networks, but also the differences of the locations where these networks sit. And so in a very small toy experiment I just looked at how big the overlap between activations of motor stimuli, of motor paradigms or language paradigms in the HCP data is, and we realized, so here the color just kind of illustrates how many out of all of the subjects have an activation at this particular position, and you see that a lot of the variability that we see is actually caused in part at least by, well, something happening, but it's just not happening at the same place on the cortex. And so I think it's worthwhile to think about the possibility that what we are talking about when we are talking about correspondence between individuals, that is not a one-to-one mapping where we have one relationship between the brain of one individual or the other individual or between groups, but it's actually a bundle of relationships that might disagree with each other, and where one correspondence might be established by anatomy and another correspondence might be established by, for example, function. And so in the next couple of minutes I'd like to show you a couple of examples how to do that with functional connectivity, but I think there are many other sources that make a sensible candidate to be anchors for correspondence, and where it might be interesting to look whether they agree with anatomy or with other correspondences. So we looked into using functional connectivity networks as a means to establish correspondence, and there is this beautiful line of work initiated by Daniel Margulis, who came up with the idea of, well, if we have these representations of functional networks and we can embed them and kind of spread them out like maps, that these maps might actually carry meaning and might be linked to the way we understand the processing in the brain. And Julia explained a lot more advanced work that was happening on top of that. We realized that these representations make sense, that they are rich, that these representations reflect some of the understanding of the architecture and the organization of the cortex, like for example the gradient between unimodal and heteromodal systems. But what I think is also interesting is that once we are able to extract them not only from groups but from individuals, they might be a good reason or a good kind of basis to establish correspondence. So this is one of our very early results of work that was mostly motivated by trying to track reorganization processes in tumor patients. And the insight was that when you do these embeddings, when you do this embedding of connectivity, for individuals that the resulting representations are stable and similar enough so that you can do registration in this embedding space, that you can establish correspondences based on how different points on different individuals, how they share maybe similar connectivity patterns. And when we look back on the cortex and say, well, okay, we have aligned these embeddings, now what does that mean on the cortex? This is one attempt to visualize that. So what you see here is an aligned embedding space, and we projected just the first, second, third, and fourth gradient back to the cortex of individuals. And what you see is that in these three individuals, the large kind of course layout is fairly similar, but you see that there are small differences between these individuals. And now we can use that in order to probe these groups for a disconnect between the correspondence established by anatomy and the correspondence established by connectivity. And we can view it also as kind of a refinement of the registration, where we say, well, okay, if we treat the aligned coefficient vectors of individuals as features on the cortex, we can kind of align, and we can constrain this alignment to diffeomorphic alignment. We can align the cortex of individuals based on these feature vectors. And I think thinking about it as alignment is one good way, and then it falls kind of in line with the concept of MSM and work by Emma Robinson. So this is work by Karl-Heinz Nenning. But I think another way to thinking about it is that this is actually helping us to understand discrepancy between these two correspondence relationships. One kind of reasonable sanity check is, of course, if we register one individual to itself, what is happening in terms of disconnect, and hopefully not a lot. So this is a color map that shows when we, over I think 100 individuals, matched first and second scan, what happens in terms of disconnect, and you see that it is fairly low. So we are in the region of about 1 or 2 millimeter, which is kind of fairly reasonable and is essentially the noise that we have in the data. Now what happens when we map between individuals is the following. We suddenly see kind of a landscape of different areas in the brain having different levels of disconnect between the two. And you see a little bit kind of mirrored in this map, the maps that you know from Sophia Müller's work, from Randy Buckner's work, about variability, but also about the shape of the networks. And so I think two things are worth kind of considering. One is that we are able, with this kind of decoupling of correspondences, to disentangle sources of variability in cohorts and populations. We can talk about, well, this is variability in the location where function is sitting, and this is variability of the actual function. And I think that helps us to better understand different effects that we are observing in populations. The second question is, does this amount or the grade of disconnect actually tell us itself something about the organization or the relevance of different features of the cortical organization? I think that is well, there are many answers. I don't have many questions I don't have the answer to, but I think this is, I think, a very worthwhile direction to go into. What is the disconnect and what's the level of disconnect at different areas of the cortex? And so, yeah, so it reflects pretty much what we saw before in the task-based experiments. And in the remaining couple of minutes, I'd like to show you two examples that go a bit closer to understanding and exploiting this idea of different correspondences. So the one is work mainly driven by Karl-Heinz Nenning and Julia Furtner, and it is about understanding reorganization processes and changes in the functional architecture of individuals who suffer from brain tumors. And so the first result we had was that when you kind of trust the anatomical registration in order to find, for example, language regions in tumor patients, of course that fails because the anatomical layout of individual positions of functional areas completely changes due to the lesion. And so what turns out to be the case is that we can use this functional space to register and to establish correspondence between a healthy control cohort and these patients and find the locations where these areas have wandered to, because the connectivity profile kind of remains stable enough in those individuals where reorganization worked. So we can use this space as an alternative kind of path to finding corresponding regions. Another way of looking at it is that we can look at this disconnect as kind of a sign of anomaly. And so here, this is a glioblastoma patient, and you see a fairly focal lesion in the brain of this patient. And if we now look at the connectivity profiles and sample it across a large control cohort and then ask every pixel, every voxel in the brain how much it deviates from this cohort, we get something like an anomaly map. And so what you immediately see is this anomaly map is not at all focal or not at all kind of centered around the lesion, but it's very, very widely distributed across the entire brain. One thing that you can get out of that is that you have a kind of a quantitative marker of the effect of the lesion, of the treatment, of surgery, of chemotherapy on this connectome, where we see kind of the quantitative changes that make up the progression of the disease and the effects of the treatment. It turns out, and I think that's a very exciting initial result, that some of these changes, some of these anomalies actually precede the recurrence of tumor, and so I would recommend to go to Karl-Heinz's poster on Wednesday to look at that. But the second question is, of course, why is it so distributed, or is there any handle on understanding this distribution? And so there, this reference space, this alternative embedding space, turns out to be a helpful kind of framework to look for neighborhoods. And so while the vicinity in anatomical space is not very helpful in predicting which systems, which functional networks will be affected by the lesions, the vicinity in the embedding space, the vicinity in the signal space of the functional connectivity itself is actually a very good predictor about where anomaly might happen. And so I think having a different reference spaces to think in and understand that sometimes the distribution and the wide kind of chaos in what we observe is just can be solved to some extent or can be understood to some extent by a switch to a different reference space to think in. I think that's worthwhile, especially if we are looking at patients where things deviate and where things kind of decouple very heavily while the brain reorganizes due to disturbance. The second example I'd like to show is work driven by Ting Xu and Karl-Heinz Nenning together with Michael Milham, and it's about the question, well, if we can talk about correspondence, can we also talk about correspondence across species? And it turns out when you do this embedding, when you represent a functional network in humans, and in this case macaque monkeys, you can actually map them and match them in this embedding space fairly well. So this is a visualization, and Ting will give a talk on Thursday on that, about aligned embeddings of the connectome of humans and of macaque monkeys. We've highlighted a couple of the core regions where we see that these regions are fairly close by in these two spaces. And then again, when we use these spaces and say, well, we trust this as an alternative source of correspondence, we can then again go back to the cortex and see where these gradients land. And once we align them, we can see that they kind of describe a very different but kind of corresponding layout of the functional systems on top of the cortex. What that means is that now again we can talk about the discrepancy of those two, but it also allows us to kind of deal with the fact that evolution affects the shape and anatomy and morphology and the function maybe in a kind of decoupled ways. And even if we are in the end finding out that they are not decoupled, we at least for the sake of observing them without bias, we have to decouple them in order to understand their relationship to each other. And what turned out in this study, one of the results was once you are using this embedding space to establish correspondence, you can essentially get rid of or can decouple the change in morphology and the change in the layout where functional systems are sitting on the cortex from how they are changing. And so if you basically remove this variability in the location, you can come up with or can observe very fine-grained gradients in preservation or change of functional systems between these two species, which I think is very exciting insight. And so to conclude, I think it's worthwhile and has merit to think about correspondence as not a one-to-one relationship, but as a bundle of relationships. And kind of disentangling these relationships helps us on one hand side to disentangle them during observation and enables us to talk about different kinds of variability that happen between individuals, between cohorts or between species. But on the other hand, the amount of disentanglement, the amount of decoupling that we can observe itself is, I think, interesting and well worthwhile observation that we can make when we are trying to understand the organization of the brain. And with that, I'd like to thank many, many collaborators and you for your attention. Thank you.